mitochondria has the membrane potential to produce ATP by oxidative phosphorylation. And membrane potential is also essential for protein import. So that without membrane potential, the matrix protein cannot go into the mitochondria. So this is the fundamental principle of mitochondrial protein import. But in 2008, very interesting paper came out from Richard Yu's group at NIH in the United States. So they found that the cyclic protein called Parkin, which doesn't have any mitochondrial targeting sequence, actually is selectively recruited to the damaged mitochondria and triggers the elimination of damaged mitochondria by autophagy. I'm very interested in this process. Why mitochondria without membrane potential recruit Parkin? So I want to know the molecular mechanism of Parkin translocation. To keep cell homeostasis, so synthesis of new mitochondria is of course important, but the degradation of bad mitochondria is also important. Okay, so in 2008, Richard group identified that Parkin is the essential factor for mitochondrial elimination. And 2 years later, in 2010, Richard Yu's group, Mazda's group, and the other group independently identified that PINK1 is also essential and actually function at the upstream of Parkin translocation. So, and surprisingly, Parkin and PINK1, both proteins have been identified as a gene product mutated in the genetic Parkinson's disease. So Parkinson's disease is one of the most frequent neurodegenerative diseases. Several papers strongly suggest that the accumulation of damaged mitochondria in the neuronal cells causes the Parkinson's disease phenotype, and this dysfunction leads to the cell death of dopaminergic neuronal cells.